If you need the link to the promotion flyer for the course, please let us know. We'll share it with you. Once someone fills out the form, we'll get a notification and be able to contact. So Transcriptomics 1, the program will leverage online materials that we offer for free, which you can sample and get a sense of the content. However, the program will offer greater detail and more focus on practical application, as well as provide guidance and community to support your learning inside the actual program. As an example, let's start with Transcriptomics 1, which is an introductory course for next-generation sequencing data analysis to study RNA expression. Let me show you how the online courses are structured. When you first get to the page, you will see that if you are logged in and approved member of the program, the course will be free. For those who are not a part of the program, the course is paid and it actually requires a membership. Once you select Take This Course, you'll see that each course has an overview video. These are course topic overviews, and these should be under 10 minutes long. Online learning experience is designed to track user activity, as well as help evaluate the efforts and the practical steps that are going to be taken. These are visible to the students, which are you, and then the program members and faculty. Once you enroll in a course, you'll see the course materials, and these are organized in course sections, lessons, quizzes, and assignments. Each course is going to have a control section at the top. These include your profile link, and then points for your activities, and they will also display recent notifications. If you look at the top of the screen, you'll see the orange section of the control bar. You can find additional icons. These controls will simplify navigation around the site and help you find the relevant materials you need for each lesson. Excuse me. The lessons on the site can be downloaded as a full PDF, and in these, they'll be available as an individual session. They have the same content as the online material. However, when you're online, you get more interactivity options. You can track the course progress each time you complete a lesson. And then this progress is going to be updated as well as visible for you and any faculty. Note that there's an online glossary of terms throughout the lessons, and these will be hover over things that you'll be able to read in addition to the material that's inside the lesson. The course also includes practical assignments that can help you practice what you've learned. These will be timed, but they have longer completion timeframes. They are scored, and they will include practicing bioinformatics analysis on our server. The URL for that server is server.t-bio.info. If you need a link to that, please let us know, and we will send out that in the chat. It's important to have 2 logins for each user. You're going to need to have one for the educational portal and then another one for the analysis platform. Let me send you that link real quick. It's going to be server.t-bio.info. There you go. Okay, so you need to have a login for both the educational portal as well as the platform. And then inside the course itself, we are going to explain the steps you'll need to do in order to perform a demo analysis. In the assignments, you'll be running a full pipeline and have to understand the outputs. Once you complete your assignment, you need to submit it, and it will upload to the site. And at this point, we will receive an email with the details of that submission, and then we'll evaluate those assignments. The platform is going to provide you with a profile where you can find your activity and learning progress. For each of your courses, you'll see submitted quizzes, assignments, and the pipelines that you have completed. After you've completed the course, your activities will be graded, and you'll receive a certification if the criteria for that has been. So let's talk a little bit more about the OmicsLogic program. In the program, we're going to focus on one type of data, which is transcriptomics. The program's going to cover analysis of gene expression data, and it's going to have practical sessions and online tutorials. If you have a basic knowledge of cell and molecular biology, you should be able to conceptually understand and then practically apply standard data analysis tools. The goal here is to process, analyze, interpret complex transcriptomic data. We'll be doing so by leveraging regular online webinars, practical exercises, and online courses. The resources are going to provide a broad but yet detailed overview of the processing, statistical analysis, machine learning tools, and single-cell data analysis techniques. We're going to cover methods, expand on various terminologies, provide practical examples, and then we're going to offer quizzes so that you can evaluate your progress. In addition to the courses, you'll be getting access to hands-on exercises. These include data sets for practice. They can be completed without any coding knowledge, and you're going to be using our tBioInfo platform. It provides an interactive learning experience. The platform's color-coded, and this is all done to establish a logical connection between theoretical aspects of data analysis methods. The practical steps needed are going to be outlined to achieve a biologically interpretable result. And so this is an example of one of the pop-ups that will occur during your experience with the demo pipeline. For these are going to be offered as tables, graphics, and interactive visualization. These are meant to help you understand the way data can be used to understand a biological phenomena. This is an example. And then here we'll see how those link to the genome. So a major topic of analysis of transcriptomic data is the identification of differentially expressed genes and isoforms. This is a straightforward approach that allows us to compare groups of samples under different conditions. We have to make several considerations around this data preparation, such as normalization and statistical adjustments. And then in this session, this is where we're going to expand on those methodologies. The online session will supplement information that is offered in the Transcriptomics 2 modules. It will describe a number of statistical techniques that are typically used for simple and more complex approaches that are going to be used for finding differentially expressed genes. I'm going to go ahead and pass the presentation to Ilya Brodsky. He's going to talk briefly about the data science portion of the program. Ilya, are you there?
Yes, I am. You have to stop sharing for me to share.
Okay.
Okay, great. Can you see my screen? Okay. Can you see my screen?
I can.
All right, great. Thank you, Joe. So we will talk about several data science aspects that have to do with specific type of data that sequencing produces for studying expression of genes. For example, one of the topics that we will focus on is how to perform different transformations and exploratory data analysis to the data and understand how we can extract relevant signal from the noisy data that RNA-seq represents. We'll talk about log-scale transformation. We'll talk about what exactly it is and how does it help us study our data and extract better signals. We'll also talk about how to use different tools. Some of them will be standard tools like Microsoft Excel or SPSS, and also R and programming packages that will help us extract the data and see better what we can study. Now, when we come to the analysis of gene expression, this session will actually help us understand the data as a whole and then to identify specific types of patterns in our data that are relevant to a phenotype. So in this session, analysis of gene expression, we'll talk about unsupervised machine learning. So for example, PCA, different clustering techniques, and we'll take a look at how the clustering techniques help us extract gene expression profiles that can differentiate between different samples. When it comes to exploratory data analysis, tools like PCA or T-SNE help us reduce dimensionality and focus on the most informative properties of the data. For example, with PCA, we'll learn about variance between samples, and then we'll talk about how we can explain this variance using gene expression. We'll also talk about principal component analysis of the samples, as well as principal component analysis of the individual genes to help us understand the gene properties as they show us the variance between samples. As a result, we'll take a look at the outputs from principal component analysis under different conditions. We'll use several examples to illustrate how to interpret the variance between samples. So here you can see an example using breast cancer cell lines that help us differentiate between the different subtypes of breast cancer. Then we'll talk about data mining in more detail. When we talk about data mining, we're talking about having a large data set. This large data set contains thousands of genes and maybe hundreds of samples. And our objective is to mine this high throughput data, this rich data set, to identify patterns that might be interesting. So machine learning typically can be divided into supervised and unsupervised. And we will use both techniques to first understand the data, but then also to classify the data based on some understanding of the data. For example, if we found a number of different subtypes of breast cancer, for example, there are 4 or 5 and different methods identify different subtypes, then we can use that to, for example, train our model using decision trees or random forest and other types of classification tools. And also we'll talk about different issues that come up, issues of reproducibility, overfitting, and generalizability of the models that we train. So here you can see an example of a gene profile that displays a certain pattern, and the question will be, well, in this example, how can we differentiate between, for example, basal, cloud and low, luminal, and other types of breast cancers that we are studying. And as an example, we will leverage some of these classification techniques to explore more in depth the other types of classification tools like discriminant analysis and support vector machines that can help us make better classification, better quality or better accuracy of classification using higher dimensionality. At the same time, we can use feature selection as a part of, for example, a random forest, where we can evaluate feature selection in a step-by-step manner, and then we can identify those features that are most useful for separation between the classes that we have been using. And that will increase the efficiency of our model prediction and accuracy of the model prediction. But also, it is going to be used for visualization. But at the end, we're not talking about just statistical analysis. We're actually talking about biologically interpretable results. And for us to be able to interpret the biological meaning of the data and of the different features of the data that we're studying, we have to be able to annotate the genes that we found and the gene sets and associate them with gene function, protein function. The genes encode for protein, so we have to look at gene ontologies, as well as pathway analysis, and also how the statistical significance of pathway enrichment can help us identify not only statistically significant biomarkers, but also biologically meaningful biomarkers. So for that, we'll try and use gene set and pathway enrichment, using pathway enrichment analysis using pathways that are found on KEGG. So here you can see differential expression. The genes that are upregulated are in the red. The genes that are downregulated are in the red, in the green. And you can see it mapped on a pathway that is statistically significant because it is enriched with genes found in the data set. Single-cell transcriptomics is a big kind of emerging aspect of RNA-seq. Up until now, we've been talking about bulk RNA sequencing, when we sequence a whole sample that might contain a number of different cell types. For example, when you capture a tumor, within that tumor, what's called tumor heterogeneity. You have maybe some metastatic cells, maybe some cells that are not metastatic. You might capture epithelial cells and other types of cells. You might even capture some of the microenvironment. But when you're studying the bulk of that data, you're extracting the most prominent signal. At the same time, when you're able to separate those cells into different subtypes, you might get a much better understanding, for example, of a proportion of how many metastatic versus how many non-metastatic, how different variation in gene expression within those cells can play a role in your ability to, for example, diagnose or prescribe certain treatments. So we'll talk about different types of single-cell transcriptomic techniques, which, by the way, over the years have emerged, and now we have quite a number of different techniques for single-cell sequencing. Now, single-cell data is very different from bulk RNA sequencing. We talk about separating cells into different types of cells, but we also talk about different ways of looking at the data, which is what's called zero inflated and has a lot of zeros, and very noisy because we have low coverage for different types of cells. So instead of bulk RNA, where we have PCR amplification that helps us study the most prominent expression levels, here we have to deal with non-amplified and low signal and still extract some meaningful population data for this type of sequencing. At the end of the program, one of our focuses is to allow independent project work. So after you've learned how to practically analyze the data, how to look at different types of data, bulk RNA, single cell, and other non-coding, poly-A, non-poly-A, so different types of RNA-seq data, you'll be able to select a project from one of the projects that we offer, or propose a project of your own following some project preparation process that will help you get the most out of the project. We prepared a number of projects that you can start with that cover different types of next generation sequencing aspects, from cancer to bacterial infections to viral and even rhizobacteria. So we'll take a look at different examples and we'll provide you with guides, and you will be able to either work with one of our mentors, or you'll be able to work as groups as a part of the program, and we'll guide you step by step on how to select the data, prepare the data for analysis, structure the data so it's ready to be analyzed, and then ask the right questions, questions that will mean something significant, something different from what's already been done, and then to, of course, present your results either to your peers or at a conference or at a poster session that you might have access to. So working on a project is important because you'll understand the details of the methods that we reviewed. You'll really see how they play out in a real setting when you have to deal with a real data set. And you'll be able to see how to apply RNA-seq not to any data set, but a data set that you're interested in and a data set that you have found to be engaging for you and for your community. We also have examples of students working together. So here you can see a group of students that we've been working with at Loyola University that went through the program that we describe here and apply these techniques to a data set that their professor has selected for them, and then presenting those results to the peers. Obviously, people start asking questions. You have to respond to those questions. So that allows you to really prepare and think through the reason why you selected certain steps and what is the outcome in terms of its biological meaning. In the past, students that have participated in our programs have went on and presented their results at different conferences and poster sessions, and have gotten their posters into conferences on bioinformatics, neuroscience, and other types of research. We do have a lot of participants that have finished programs in the past. We don't only do programs on transcriptomics. There's a variety of OmicsLogic programs that we conduct throughout the year. And students at an undergraduate, graduate level, but also faculty have joined these programs in the past and have found the materials that we prepared to be both comprehensive and engaging both for students, for faculty, and for researchers. So we don't narrow the target audience here to be just undergraduate students. We think that anyone with an interest in RNA-seq, in analyzing data and interpreting that data, can benefit from the program, because the program is structured both by online self-guided materials that Joe explained very well in the beginning, and interactive sessions like this one, where we go through and guide people to use different methods. You will be able to both gain a practical skill of analyzing data and a theoretical, more research kind of overview of the different methods that are typically used for the analysis, as well as the different types of projects where RNA-seq could be a useful technique. So we encourage you to explore the program. The program is going to be starting in January. Let me just go here quickly to the link. So the link can be found right here. I'm going to just paste it in chat for anyone that would like to join. And here you'll be able to see some overview of the program topics that are covered. There's a video that describes the topics in greater detail, a schedule starting from January 14th, which is next week. And here at the end, if you are interested, you can fill out this form and submit it. And after you fill out this form, you will be directed to complete your registration. So to complete your registration, what Joe showed in the beginning, you will see some of the online course materials that we have, and also the platform that we will use to analyze data. So just to briefly show you what the platform includes and how it is used, I'm going to switch over to the screen. Once you have your account, you can, by the way, request a demo account to see some of the elements that we have here. We have a whole section on next generation sequencing data that includes transcriptomics, genomics, epigenetics, and other types of data, including metagenomics. But we also offer sections on mass spectroscopy, structural biology, data integration and modeling, and then a lot of different tools on data association and data mining. And we're always developing new sections, and right now we have a section on virology, cancer biology, and biology of aging, and those are in development. By using the platform in the course and the practical sessions, you will be able to try out different types of approaches. For example, for RNA-seq, for transcriptomics, we will use a couple of projects. One on cell line projects, where we have a collection of different cell lines from different types of breast cancers. And we have a PDX, a patient-derived xenograft model project, that you can play around with and understand the data. So we explain here every step, and we also provide the technical description of the methods, as you will see in the following steps. A typical RNA-seq pipeline, a processing pipeline, will include several major steps, like pre-processing, where we have to trim the adapters and remove the PCR amplification. And then we can map our data using a variety of mappers, HiSat, Bowtie, and others. After that, we have to quantify the data. And for quantification, there are also a number of different techniques. So for example, using HT-seq. And once you prepare this pipeline, you can quickly see the results. And so once you see the results, once you understand what the results look like, now you are ready to use the platform without a demo. Once you are ready for that process, you'll be able to upload a data set. And here you have to make some choices. For example, the PDX project is a pair-end project with FASTQ files, and you can map it on both the human and the mouse genome. Once you upload the data, you will be given the data set that you will pass here. here you can add more. And now you have to do the same steps to actually run the analysis on the data that you prepared. You can, of course, select the most appropriate type of analysis for what you need in downstream steps. After you have prepared your pipeline, you'll be able to save the pipeline and run it. In some cases, you would want to transfer the analysis from the platform out of the platform and do it in R or Excel or other types of environments. What you can do, for example, is analyze the data. A lot of the techniques right here that do statistical analysis, annotation, and data mining techniques, they will provide you with an output file that actually starts with a script. And then on the platform, on the educational portal, you'll be able to take that script and go through steps to modify the script to, for example, produce a visualization that is more informative and more relevant to the type of analysis that you have done. So just an example of one of the courses that does that is going to take the output from principal component analysis and guide you through the steps of how to modify the script in R and produce a different type of a visualization. So here you will learn about the R coding environment and how to use the standard outputs of that script, and then also how to modify that to improve the clustering and visualization and make the visualization more informative. So briefly, this is an overview of the program. The topics, like I mentioned, are going to be available right here, where you can see the different sessions that we are planning and the topics that are going to be covered. We hope that you can join us and go through some of these materials together with Joe, Mohit, and myself. We'll be taking different sessions to talk about different parts of the program. And I'm happy to answer any of the questions that you might have, either about the program, about the platform, or about RNA-seq itself and transcriptomic data analysis.
We would request participants to unmute themselves and ask questions regarding the presentation that we went through and regarding the courses or the registration details, etc.
I do, by the way, want to just mention briefly that we have, in parallel, a number of programs that are going to be happening at the same time. And we know that you probably saw different types of emails and announcements that we've done. So I do want to just clarify what the differences might be. We will have, this program is focused on independent research projects and helping you understand RNA-seq, but it's for an online audience. There's not going to be a physical place where we meet. It's just fully going to be online using Zoom like today. At the end of the program, you will be able to receive the recording. There are other programs that are going to be available locally. For example, we have a program that's going to start at Amity University in Kolkata in India. So anyone who is in India, and primarily this is for students from Amity University there, but I believe others might be able to join that as well. You should learn the same elements of the program that we are talking about today, but in that program. And then we have a course that is a graduate course at LSU, and this is in Louisiana, and this is sponsored by the Louisiana Biomedical Research Network. So if you're a part of any campuses of LSU or any other universities in Louisiana and you would like to join that course, that would be a better course for you to join because it will have sessions that are on-site and they provide you Q&A and interaction with faculty from LSU and from the Louisiana Biomedical Research Network. There's also a program at Georgetown University in Washington, D.C., that will cover aspects of next-generation sequencing data analysis for genomic data and transcriptomic data. So if you're in DC and you want to learn about those, you can reach out to us about that one as well. And then we, of course, have programs that are not just focused on RNA-seq, they're focused on many other types of data. For example, genomic data, transcriptomic data, metagenomic data, epigenomic data, and others. And after this program starts, we will start sending announcements and posting them on the site as soon as we have the dates and the topics confirmed, which is probably going to start after the 15th of January. So please make sure that you're following those updates as well. Okay, so question, why are we going to use R and not Python? Yes, it's true that Python is often used for data science and machine learning, but in R, there are a lot of packages that make it easier for biologists to use a single package that does many things: statistical analysis, annotation, visualization, etc. So that's why these programs are actually primarily focused on people that have a background in life sciences. So they probably already understand some of the biological background of the project, and they don't have a lot of experience using coding. Python does require a little more background in coding and a lot more background in biostatistics or machine learning, and that's just a lot to combine in a single program. In the future, we hope to offer a lot of the same elements using Python as well, but this specific program is going to include just elements of R, and a lot of those are going to be optional. So you can conduct all of the analyses that we will discuss, not doing any coding at all, but you will have a chance to try some of the packages, right? And packages here is key word because bioinformatic packages are abundant in R. And for Python, of course, you have BioPython, but it's not as easy to just get started with. Okay, an independent research project can be used for further research. I mean, how much authentic this independent research can it be presented as evidence for further research in Research Institute? Definitely, yes. So, you know, as I showed before, we had a number of groups, and maybe I can just, you know, point out some of those projects that were done. We had a whole team of students, international students. One of them was from the US, one of them was from Germany, and a number of them were from India and Pakistan. And they worked together on a project on Alzheimer's disease. They also did a project on neurodegeneration, the basic biology of neurodegeneration by looking at different types of neuronal cells. And then they presented their poster at the annual Bioinformatics Research Conference at Louisiana State University. We also had a student from Israel that did her work on a genetic disease that leads also to neurodegeneration. So this was a neuroscience program, and she went on to do her PhD using that data set. We also had students from Tulane University that worked with their faculty. And like I mentioned, we had students from Loyola University where they worked on a project with arthritis data. And later on, the faculty member actually took a sabbatical and continued working with that data set in her sabbatical. So I think that, you know, it obviously depends on where you are now and how much clarity you have on the data set that you have chosen to work on and who is going to be your partner. But that's what we hope to achieve with this program. With this program, in the past, we've had very good interaction between students and faculty. Actually, the professor from Tulane University, Ian Townley, ended up working with a partner from the UK to analyze some of the data that he had sitting from his lab. So I think a lot of this actually leads to research projects, and the research projects are going to be informative for research discussions as you plan your analysis further. You're from plant biology background. Is there hardcore mathematics to understand? So we try to avoid formulas as much as possible, but when we do speak about formulas, because they're hard to avoid, we do explain them in language that I think biologists can understand, and we try to find examples that clarify the mathematics behind it. I don't think that you need hardcore mathematics or biostatistics to take this course and get something out of it. For those of you that have a plant biology background, I suggest to take a look at one of the projects that we have, which is actually on the impact of drought stress on potatoes. So you can find this project right here. Of course, right here, it's mentioned as paid. If you're a part of the program, it's going to be accessible to you as well. But what I'm trying to say is that we already have examples of projects. And by the way, this project was prepared also by a student. The student was from University of Nebraska, Omaha, where they study a lot of plant biology as well. But these types of projects are also using a lot of RNA-seq to understand the molecular regulation of different stress response. And there's a lot of other topics as well in plant biology that I think could be relevant to your research. Okay, if you have any other questions, again, you can, and Nitin, if you can let me know if that clarifies your question, I can clarify more if needed. And if anyone else needs, if anyone else has any questions, you can go ahead and unmute yourself or even paste it in the chat like the others did. Are there day-to-day classes? So what I suggest to understand the program schedule is to look right here. On this link, you have the list of sessions that we have. As you can see, they're once every few days. They're not going to be every day. And the sessions are all going to be at the same time, online. And if you can't make one of the sessions, you will be able to get a recorded session and then participate in the discussion. For example, post your questions on a forum or even schedule a meeting with one of the trainers online. Okay, yeah, thank you. Will a similar program like this be available in the fall? So Tara, I don't know, which university you're from. So we do have a schedule for online programs throughout the summer and in the fall. So definitely, yes, online you'll have those. But if you're in, one of the areas where we have a collaboration with a university, like if you're in Louisiana, if you're in around DC area or other areas that we have something going on, I'd be happy to connect you with those programs. Can you let me know what university you're in? Tara? Okay, great. So for Louisiana University, so Southeastern Louisiana University is a part of LBRN, and they will have an undergraduate course starting in the fall. They will also have a summer course. And depending on your level, we have an undergraduate course, or there will also be a graduate course that will be handled by LBRN and available online to all the LBRN campuses. So if you write us, I'll be happy to connect you with the right information on that. And what I also would suggest is looking at this link. So this is, these are the programs that we run for the Louisiana Biomedical Research Network. It's focused on Louisiana universities and campuses. And here is the upcoming LBRN transcriptomics program. It's a very similar program, but it's a graduate course. Undergraduate students can also join this, but of course you're asking about the fall program. So after this program is over, you'll be able to see details for the summer and the fall programs. Juris, one sec. So let me just paste this for Tara. So you can take a look at that. What can you recommend for someone who is coming from a pure computer science background, 10 years of professional programming with some biology? Do you think your platform can provide that introduction to the biology part of bioinformatics? Do you think your platform can provide that introduction to the biology part of bioinformatics? Okay. So yes, I think that that's a major challenge for computer science, people, students, researchers, etc. You probably have a lot of technical know-how and familiar with a lot of different approaches. But one of the major questions is, okay, how do I select a project that is going to be meaningful for me? How do I conduct the analysis in such a way that I don't get some interesting patterns, but I can also interpret the patterns? And then finally, when I interpret those patterns, how do I pick some kind of a topic that is going to be impactful and interesting? And that is a big part of the program that we try to incorporate into the sessions, but also into that portion on the independent analysis. The platform itself, the reason why this platform is useful for someone that knows how to code isn't just the coding part, right? So bioinformatics is much more than coding. Bioinformatics is critical thinking, is understanding what kind of methods are going to be appropriate for what kind of data. And then finally, how do you combine those methods and the biology to make something meaningful? So the transcriptomics program will start off by explaining the biology of RNA transcription. And it's going to talk about alternative splicing. It's going to talk about how that plays a role in a variety of pathologies and different projects. For example, how is it different for cancer? How is it different for viruses? How is it different for plants, right? You have to understand some of those basic things. Then after you understand that, you have to also understand the properties of the data itself. When we talk about RNA-seq data, these are short reads mapped onto genomes. Genomes have a certain structure. And when we try to assess the level of expression, we have to understand what those numbers mean. After we have those numbers, there's certain transformations we have to do. We have to, for example, do log normal transformation. We have to look at the distribution. We have to then analyze that normalized data. So those things will play a big role in how we interpret the results. For example, what is a high enough level of expression when we talk about log FPKM values? Why do we use FPKM when we do PCA? And why do we use raw count when we do differential gene expression? Those are questions that have to do with both the data side of things, but also have to do with the biology data, with the biology side of things. You know, when you do fold change, what is the meaningful fold change? Meaningful for what, right? So all of those questions are the questions that we will try to answer in the program. Yes, I definitely recommend going through some materials that will give you a background. And what I recommend are, again, what we try to prepare on our end are some of these free resources, right? So a lot of people ask us, why do you offer a lot of these materials online for free, but then you have these programs? Online materials, what a lot of you probably know is you try to take a course, you kind of understand 10, 15, 25% of the course, but you completed it, right? You're not going to go back and review all the details. The program helps overcome that because you have a guide. You have someone that takes you through the steps one by one, making sure that you understand them and you can apply them. And you have somebody to talk to, you have somebody to ask questions about. So what I recommend is going through Introduction to Bioinformatics that will provide you with some overview of important concepts and important directions where bioinformatics plays a major role in discovery and in research. Transcriptomics 1, in my opinion, is a fairly basic level that describes some of the elements of what we talked about today, and will provide you with a review that will, explain to you what are genes, explain to you what are isoforms, what are exons, introns, different genomes, and things like that. There's also a great course called Introduction to Genomics right here that talks about some of the next generation sequencing techniques, the DNA, kind of gives you a sense of some of the terminology, what are nucleotides, etc. So I recommend some of these things that we offer. There's also a lot of material online elsewhere. So obviously there are courses on Coursera, on edX, on other platforms that talk in general terms about molecular biology, which I think coming from a CS background, you would probably benefit from. Thank you very much. Are there any other questions that we can address today? What program would be best for someone with a strong background in biology and with wet lab experience, but with no experience in bioinformatics? So again, just getting started, I would recommend taking this course. It's a free course called Introduction to Bioinformatics. Bioinformatics is a term that is often misused. Bioinformatics has gone through a lot of transformation over the past several years. When we heard the word bioinformatics probably 5 or 6 or 10 years ago, you would think sequence alignment. You would think, how to search for how to do blast searches, how to, use the site NCBI. That was bioinformatics back then. Today, bioinformatics is big data science. It's data-driven research. It's how do I come to my research question from a different data-driven perspective? How do I leverage these large collections of data sets like the Cancer Genome Atlas or 1,000 genomes or other repositories that I have to just understand what they are to be able to leverage them? And also a good understanding of data science tools. How do I do exploratory data analysis? How do I do machine learning? How do I train models? How do I evaluate the training of the models? Right? So that transformation, I think, is important for anyone that is now involved in life science research, because ultimately, when you do the experiment in the lab, it plays a role in your experiment design. It plays a role in what type of data you want to collect, being knowledgeable about what the output of analysis would be to understand statistical significance. How many samples do you need? What kind of conditions can you, control and you can't control, right? So those things play a big role, and they're all related to bioinformatics. So this course goes over some of that material. Data-driven discovery, real-life examples in healthcare, personalized medicine, molecular diagnostics, treatment development, as well as some of the other topics that are covered right here. I think that, and Joe can probably add to this, coming from biochemistry background, the materials that we have in Transcriptomics 1, in all of the basic courses, so Transcriptomics 1, Metagenomics 1, Genomics 1, and there are some others, will be interesting enough for you to kind of see how the tools are useful and meaningful in your type of research. Of course, I would have to know a little bit more about your background in the wet lab. Maybe you can post it on chat. And we can suggest some other materials that might be interesting or useful to you. I would also say that, not everything is a course on the basics. There are some projects here that you can scroll through. Some of them are fully available online. Others we use in programs like this. They're not fully available, but they span a variety of different topics. And, the more we know about you, the better we can help you. Yes, and thank you, Joe, for pointing that out. What we try to do, and maybe I'll just show you a couple of things. We have over 2,000 participants on the platform, and we try to try to use our point system to show kind of who's who. And I think it's very nice to understand, what other people are on this platform and where are they and what do they do. So you can go to this link and see some of the top profiles that we have, just to kind of understand who they are, and see where they are from and what is their background. So you can click on any one of these profiles and obviously see little information about that person. Sarah, for example, joined already the Transcriptomics upcoming program. You can see her activity, you can see her profile. She's from International University in Florida. She's interested in forensic science and has some background in biomedical sciences. So you also, if you add your social media links, obviously people can connect. You can go and see their LinkedIn, for example, and connect with them. And I think that's really a big part of this program, is you also get to know a lot of people that have similar interests, that are also trying to develop their career and build up their resume, using some of the materials that we offer. And you can see their profiles and kind of learn more about these people and connect with them further. Okay, well, again, it's great to meet all of you, those that had questions. I hope that I was able to answer some of the questions. If you have any other questions about other programs or other resources that we offer, please ask us. Rocio, sorry, I just saw the message. You're working on cancer by including and analyzing potentially relevant mutations and developmental biology by developing animal models to question the relevance of some genetic elements for survival and differentiation of modern nuclear cell populations. Yeah, honestly, I think that a lot of the things that you're describing here, we're going to be in this program. We'll be going through RNA-seq for cancer diagnostics. So how can you diagnose a specific subtype of cancer? We'll be talking about differential gene expression, for example, the difference between metastatic and non-metastatic cancer. We'll be talking about, single cell data, which you also seem to be interested in. So I think that you will definitely benefit from the program that we have, and we have quite a number of projects in cancer, starting from cell lines to animal models to patient samples. So I encourage you to try and join if you can. If you can't, then at least take some of the free courses online. Juris, yes, the certificates. So again, it depends on where you are. Where are you, Juris? Sorry. Which, geography? Where are you? Are you in Europe? Are you in the US? And I will just mention this briefly, in London. Okay, so if you're moving to the US, employers in the US obviously will look at the basic background, CS, biology, whatever. But primarily, they will also ask you about projects. And that's a huge part of the interview process. Having a CS background is great. Data science is, as you know, becoming a very hot topic everywhere. But data science is different from bioinformatics. And data science or machine learning in bioinformatics is an extremely high growing area, where you have to differentiate yourself, right? You have to be able to say, what is it that you did with the tools that you know how to use? What problems have you been able to solve? And that's what this program is going to be designed around, right? You will get a certificate, and certificate is okay. But when they will ask you, what project did you work on? And you are able to think through, okay, what is the data? How much data? Analyze that in a short period of time. If you're going to be looking for a job and you want to focus on personalized medicine or neuroscience research or some kind of a multi-omics integration using deep learning and machine learning, I mean, those are the kinds of things that are going to stand out to your employer, potential employer. I recommend RNA-seq because that will start from kind of a more basic level and get you all the way through to single cell. But then in the future, what I would also recommend, we don't have it right now, but we will have another program called Data Science for Biomedical Data. And that's more of a data science application. How do you apply it to solve some of these hot topic problems that are relevant to pharmaceutical companies, clinical research organizations, research centers. And we also regularly, and I'd be happy to add you to our LinkedIn group. We regularly post some of those job opportunities and research opportunities in our LinkedIn group. So if you're interested, you'd be able to apply for jobs there. Of course, we don't do job placement, but these are just guidelines and suggestions from our side. All right, great. Thank you everyone for joining today. It was great to meet you. Again, we sent you the links in the chat. We will follow up with you. How frequently can we interact with the project supervisor during working on the project? We're a guideline. So we do require for you to get to a certain level before you start interacting with the supervisor. Essentially, you have to complete several of the online courses, and you have to start practicing some pipelines to just understand what is the data and what are you trying to do. Then there will be a form that you have to fill out, and we'll guide you about selecting the project data set, planning your analysis, thinking through the question that you're trying to ask, etc. After we get that, we will reach out to you and schedule between 3 and 5 meetings that will either be as a group or one-on-one to discuss the details of the project. All right, great. Thank you, everyone. We'll send the recording after we end here, and great to meet you again, and hope to see you as a part of the program. Take care.